This is part 4 of 5 on Chapter 9, Microbial Genetics. In this section, we're going to be looking at variation in DNA and looking at mutations. So mutations are just changes in genetic code. So any change in the phenotype, so the physical characteristics due to a change in the genotype or the genes or DNA, so it's nitrogen base sequence of the DNA, that is called a mutation. So any natural, non-mutated characteristics are known as the wild type or wild strain. Any organism that has a mutation is known as a mutant strain. Okay. So we're going to be looking at different types of mutations and how they can occur and the different terms we use to describe mutations. So isolating mutants, so a lot of bacteria just mutate on their own, and we'll get into the different ways. So isolating mutants, if you see a mutant that you want to grow, you can do what's called replica plating technique. And this allows for the identification of mutant bacteria. So up at the top, we have a tube, a broth culture with some bacteria in it, and we treat it with a mutagen. It could be a chemical, it could be UV light. We'll get into different types of mutagens in a little bit. So you put that mutagen into your culture of bacteria. You then plate that bacteria, so you let it grow and incubate it. Then to replicate, you take what's called a replica block, and it has a velvet surface on it that's been sterilized. So you just take that block, press it down onto your plate so you pick up the colonies, and then you can take your replica block and put it onto different plates. So this is down at part C. So we can have a replica plate that has a complete medium, it has all the nutrients a bacteria needs to grow, or you can also place it in a replica plate that is missing one or more nutrients. And then you incubate that plate, and you can actually identify the different mutant based on if it grows on one plate and then if it doesn't grow on the other plate. So all strains grow on the complete medium, whereas the mutant colonies do not grow on the media that's missing a nutrient. So you can then pick up that individual colony that grows on one plate and not the other. You know it's a mutant because it grows on one, not the other, and you can isolate it and put it into its own test tube or broth. So I talked about we had to treat our bacteria culture with a mutagen. So different causes of mutation. So there's spontaneous mutations. These are just random changes in DNA due to error during replication, and we don't really know what the cause of this error is. So that's spontaneous. Other types of mutations are called induced mutations. This is when we expose our organism to a known mutagen. In this table 9.3, it shows different mutagens. So we can, if you look on the agent, there's chemical and radiation mutagens. So chemicals, ethidium bromide is one, different dyes. So these different chemicals can do different things. So they can remove amino groups from bases, changing them, place inserts between different bases, cause frame shift mutations, which I'll show you in a little bit. Another type of agent is radiation, so gamma rays, X-rays, or UV light. This radiation, they form free radicals, and they can cause the DNA to break, or they can cause cross-linking between adjacent thymines. That's what UV light does. So if there's two thymines right next to each other, instead of those thymines pairing with the adenes across from them, those two thymines will pair with each other in the same strand. So these are different mutagens that you can expose organisms to change or mutate their DNA. Different categories of mutations. There's point mutations. This is where you add, delete, or substitute one base or a few bases in your DNA. Some point mutations are called silent mutations. These mutations, they alter the nitrogen bases or the DNA bases, the order of them, but it doesn't change the amino acid sequence. So this kind of goes back to the second part of this chapter when we talked about transcription and translation. If you look on that genetic code chart, I pointed out that the chart is redundant. So there's multiple codons that can code for the same amino acid. So if you have a change in your codon, but you still end up with the same amino acid, it's called a silent mutation. It didn't really change your protein that was being created. And then we also can have back mutations. This is when a mutated gene reverses back to its original composition. So we're just back mutating back to the wild type or the original sequence. So here's some examples of different mutations, and we're going to be using these sentences, these 3-word sentences, to kind of help you make sense of the different types of mutations. So if our codons are the big bad dog ate the fat red cat. In this example, we have a substitution missense mutation. So we're going to have a substitution. So in the word fat, the A in the fat we change to an I. So it's a substitution, and it's a missense because the sentence that we create still makes sense, but it's different than the original. So we get the big bad dog ate the fit red cat. So we still have a sentence that makes sense, but it has a different meaning compared to the original sentence. Another type is a substitution nonsense mutation. This is when your sentence doesn't make sense. So we have the big bad dog ate the fat red cat. And we have a mutation where the word dog, it mutated into a stop codon. So now we just have the big bad and then the sentence stops. So we just had a simple substitution, but now our sentence doesn't make any sense. It's a nonsense. So our protein isn't created in full. Another type is called a frame shift insertion mutation. So again, we have the big bad dog ate the fat red cat. In this instance, in the word bad, so in the third word, in between the A and the D, we inserted a B. So we inserted a nitrogen base. So now that caused our sentence to have an extra letter. So we have an insertion, and then it shifts all of your codons. So now we have the big, bab, DDO, gat, tray to cat. So it's called a frame shift because you just move everything down one, and then it no longer makes sense. Another one is called a frame shift deletion. So instead of inserting, we actually delete. So in the word bad, we have taken out the A. So we still have a sentence, but it doesn't make sense. Everything has shifted because we deleted one nucleotide. So these different mutations that can be caused by mutagens, or we can just have the spontaneous mutations. Sometimes the cell can actually repair the mutations. So since these mutations can be potentially fatal, not good for the living organism, the cell has several enzymatic repair mechanisms. So they have enzymes that can actually repair the DNA. And these enzymes are constantly proofreading the DNA, so they find and repair damaged DNA. And there's a few different things. There's 4 things that cells can do to repair these mutations. So there's a DNA polymerase. There's a type of polymerase that proofreads nucleotides during DNA replication. So as you're replicating DNA, what we went over in the first part of this chapter, DNA polymerase is constantly proofreading and repairing damage that occurs. There's mismatch repair. These enzymes, they locate and repair mismatched nitrogen bases that were not repaired by the DNA polymerase. So this is another kind of checkpoint that can occur. There's light repair. So if you expose an organism to UV light and they get damaged, you can expose them to regular colored light, so visible light, and that visible light can actually repair the DNA damage. And then the fourth one, excision repair. These enzymes, they locate and repair incorrect sequences. So in this instance, these proteins can actually remove larger segments of DNA. So they remove a segment of the DNA, and then they add in the correct nucleotides. So DNA polymerase and the mismatch repair, they usually replace just like one nucleotide, maybe two. The excision repair, the fourth repair method, they can replace a larger sequence of DNA. So, and here's an example of the excision repair mechanism. So your enzyme complex, the big yellow protein, attaches on, it cuts out a large section of your DNA, and then a second complex of the enzyme comes in and adds the correct base pairs in there. So they cut them out and then put in the correct ones that are supposed to be there. In order to identify different mutagens, scientists can do something called the Ames test. So the Ames test, it can test any chemical. So any chemical capable of mutating bacteria DNA, they automatically assume that it's capable of mutating mammalian DNA or human DNA. So scientists can actually use bacteria to determine if a certain chemical in like shampoo, for example, or chemicals used in food, if those chemicals mutate bacteria DNA at a certain rate using the Ames test, then they have to put a warning on it that it's capable of mutation or it's harmful to other organisms. So agriculture, industrial, and medicinal compounds, everything is now screened using this Ames test. So what we do is they use a culture of Salmonella bacteria, which is histidine negative in here. Okay, so they take this special Salmonella bacteria and they plate it onto a control plate, which is just a medium that lacks histidine. Okay. So this is a control plate. The Salmonella are not supposed to grow on it. They also plate that same bacteria onto an experimental plate that contains the compound we want to test. This is our test plate. It's the pink one on the right. So this test plate also contains minimum medium and also the test chemical that we are testing to see if it's a mutagen. They incubate the two plates for 12 hours. Any colonies that grow have back mutated, so they've gone through a mutation that allows them to grow on the plate. So on your control plate, some of the Salmonella just automatically, spontaneously go through back mutation, so you get a few colonies that grow. If you're testing media, so in the pink, if you get lots and lots of colonies that grow, that means that lots and lots of the bacteria colonies were back mutated. And if more colonies back mutate in the presence of the chemical compared to without the chemical, then we say that chemical can actually cause mutations. And then they have to go through further testing to kind of understand what is actually going on. So again, the Ames test is used to identify different compounds that can potentially cause mutations in living organisms.